The Human Genome Project was one of the largest and the most important scientific accomplishments in history. It was expected that once the complete sequence of the human genome was known, major medical breakthroughs and cures would soon follow. But deciphering the genome proved to be only the first step.
Bioinformatics is a cross-disciplinary field that draws on computer science and statistics to compile, store, analyze, and disseminate biological information.
Originally, bioinformatics was synonymous with genomics, a branch of molecular biology that focuses on the complete set of genes and genetic material in the organism. Sequencing of the human genome only revealed the linear sequence of bases in our DNA. The work of finding genes and regulatory regions within the DNA sequence is ongoing and continues even up to today.
The nucleus of each cell contains the complete genome, but each cell only expresses a fraction of these genes. Epigenomics is the study of how gene expression is regulated by reversible epigenetic changes to DNA histones, such as methylation and acetylation.
Transcriptomics using techniques such as microarrays or RNA-seq to compare gene expression levels under different conditions. RNA is extracted from a sample, and the sequenced reads are mapped to the exon in a reference genome for counting.
However, RNA levels only account for about 40% of the variation in protein levels in the cell. Proteomics uses methods such as gel electrophoresis and mass spectroscopy to identify and quantify the complete set of proteins in the cell.
The function of most protein in a cell is affected by post-translational modification like phosphorylation and glycosylation. Phosphoproteomics is a branch of proteomics focused on proteins containing phosphate groups, while glycoinformatics focus on glycan modifications.
The activity of small cells is more than just the expression of genes and proteins. Metabolomics is the study of small molecules involved in metabolism, including amino acids, nucleotides, and vitamins.
Lipidomics focuses specifically on hydrophobic, non-polar compounds such as fatty acids, cholesterol, and triglycerides.
A benefit of high-throughput methods like RNA sequence is that they can sequence a large amount of material at the same time, but tissues often contain a mixture of different cell types that bulk sequencing ignores.
In single-cell transcriptomics, cells are separated into wells and individually labeled before sequencing. This makes it possible to analyze different cell types separately.
Similarly, in spatially resolved transcriptomics, an intact tissue sample is fixed to a slide and cells are labeled based on their coordinates. This makes it possible to compare cells at the margin and the interior of tumor.
Each omics technique provides insight into a different aspect of biology. Therefore, it is useful to combine two or more different omics techniques such as transcriptomics, proteomics, and metabolomics.
The Human Genome Project taught us not to expect dramatic breakthroughs as we peer into the genome, but multi-omics is slowly revealing the hidden world within our cells.